Proteins. Proteins are made up of 3 or 4 chains of peptides. And peptides are made up of at least 1,000 amino acids. So if we looked at just kind of a little drawing of an amino acid, there are 20 amino acids. And if you join them all together, you would have something like amino acid number 12 joins to amino acid number 4 that joins to amino acid number 3 that joins to amino acid number 11 that joins to amino acid number 19 that joins to amino acid 20 which joins to amino acid 4 which joins to amino acid 5 which joins to amino acid 10 which joins to amino acid 13 which joins to amino acid 4, so on, so on, so on, so. Okay, so that long chain right here of amino acids would make up one peptide. So here we're going to say this is our red peptide. This is our red peptide chain. And then we would be able to make another peptide chain of different numbers, different orders. Sure wish we could get this to work. And let's do green. So we would be able to say here is our second peptide chain, and it's going to be number 20 joined to number 3 joined to number 4. Got the idea? So here's a long peptide chain in green, and it's going to curl up and look something like this right here. And so there is our green peptide chain. Then we'd be able to make a third peptide chain. So let's make it blue in color, and we're going to say it is amino acid number 8, which is joined to amino acid number 10, which is joined to amino acid number 11, which is joined to amino acid 4, which is joined to amino acid 1, which is joined to amino acid 13, so blah, 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 blah. So here we have our blue peptide chain, and we would make it look something like this right here. And so this structure here, that whole entire structure is a protein. In the stomach, where it is acidic, you have hydrochloric acid. In the stomach, this protein here is going to denature so that you end up with your blue peptide chain separate from your red peptide chain, so they would not be bonded together anymore. And then you're also going to have your green peptide chain off here to the side a little bit. I think I made it a little bit different on that, but you get the idea that all 3 of your peptides are going to separate from each other, and so you're going to have 3 individual peptide chains. This is going to be called denaturization. Denaturization is going to be caused by a low pH or acids, an acidic environment, or from high temperatures. So whenever you cook something like an egg, remember that it's clear and then it's going to turn white as you cook it. It's because the proteins are denatured. Okay, there are 20 amino acids. 9 of those amino acids are essential. Okay, what does it mean to be essential? It means that the body cannot make this nutrient. If it does make it, it doesn't make it in a sufficient amount. So it must be obtained from the diet. So there are 9 essential amino acids. Some books say 8, but our nutrition book says 9, and so that's kind of what I'm going to go with. Actually, there are 10 in infants and children, so kind of keep that in the back of your mind. But for our number for our class, we're just going to go with 9 essential amino acids. The structure of an amino acid. All of the structures of the amino acids have a whole lot of similar to each other. Let's erase all this so we can draw it. An amino acid is actually a pretty small structure because it's going to be absorbed across the intestinal lining. Should have got a bigger eraser. I think that's enough room here. Okay, so you have nitrogen bonding to carbon, which is bonding to carbon. You have this carboxylic acid group here. You have this little hydrogen here. Nitrogen has 3 bonds, so we're going to fill in those spaces with hydrogens. And this first carbon here is going to have an R group attached to it. This R group, if you look on the periodic chart, there's no such thing as an R. So the R group is basically that unknown. It can be something as simple as hydrogen, or it could be a carbon binding to 2 hydrogens and then binding to a chain of carbons. Anytime you see the nitrogen, you know that it's either going to be a nucleic acid or an amino acid. And since we don't spend our time in this class, we don't do a lot with nucleic acids, you can probably pretty much guess that it's an amino acid. Whenever this nitrogen group is removed, that's called deamination. It's going to be removed anytime you're going to use this amino acid for something other than making a protein. So example would be if you're going to make energy from it. The nitrogen group does not have any function in making energy, and so the amino acid is deaminated. This nitrogen group here becomes ammonia. It goes into the bloodstream, and it is going to eventually make it to the liver. The liver is going to convert it to urea, and then the kidneys are going to be able to filter it out. So if someone has kidney problems, they're probably going to have they're probably going to have high amounts of urea in the blood. If someone has liver problems, they're probably going to have high ammonia in the bloodstream.